This session is on climate change, which concerns all of us, as His Excellency already alluded to. Therefore, my topic will be within that region, but it will concern mostly on the plant side. And the title is Crop Drought Tolerance, What Can We Learn from the desert plants that we deal with in this country? Everyone knows that we are in a country where we have an issue with the desert or the increase in temperature, although you may not feel it compared to other countries, for example, in the Northern Hemisphere, as they don't get as hot as we do here. But any change in temperature, we certainly feel it, and the plants certainly know about it. Not only that, but also we sometimes feel the change through the cold winters or the cold months when we have a snowman that is Omani, like this one in 2017 in Jebel Shams. So this will be, I'm sure, talked about in further details in the coming presentations. For this reason and for many other reasons, we are as well exposed to the outcome or the problems that are associated with climate change, as well as all the adaptation and mitigation mechanisms that has to be in place. And I'm sure different organizations and many parts of this country have already started working on adaptation and mitigation of climate change. However, when we come to the ecosystem, especially the vegetation part, since this country is classified as mostly desert, which is 75% of the country, basically, we are not there on the map, actually, in comparison to other countries on the world scale, for example, in the Amazon or North America or Europe, for example, or Australia. The impact is not as much because according to the international studies, including those from the ones from NASA, the ecological sensitivity of climate change on this part of the world is much less than in other countries due to the high climate stability and low level of vegetation, and that's basically why it is. So is it a hoax? Luckily, we have done away with that story starting 2021, hopefully, but it is not. Even though we don't see the desert as an issue, we are actually living in desert, and we have opportunity actually to teach other countries on how to best use the resources that we already have. And obviously, all the studies have shown, and this is the one from Sharabi in 2013, and this has been also updated, and we were, as a university and the Water Research Center and colleagues from the college also were part of this. The studies that have prepared the impact of climate change on Oman. Certainly there are predictions on the changes related to water, and this is the title of the conference, and that's where, especially, of course, there are also other factors like temperature and many others, but the focus here is on water. And believe it or not, largest part of the country, about maybe, I don't know, 70, 80 years from now, will have plenty more rain, especially the central and southern part of the country, more than we have now. However, the center of diversity of plant species, especially the main ones in Hajar Mountains, will have much less water than in terms of rainfall compared to what is having now, or the precipitation that's going on now. So this is showing the average rainfall in the past few decades. And you can see there is a large amount of rain that happens actually in the north compared to the south, even though we get 3 months of wonderful Kharif season in Salalah. Still, the amount of rainfall we get in the north is very important to the plant diversity for the farming system, food production, because that's where most of the agriculture is happening. So what's going to happen? We are expecting many things to change, including increased desertification, increase the need for water resources, for irrigation, so many consequences for desert farmers. But this is not something new that we have dealt with. Our ancestors have already had this problem before, and they have dealt with it, and successfully for thousands of years. The first time they got here, I'm sure there is not plenty of water. As I have mentioned, we are classified as a desert country and desert arid climate, and therefore they invented ingenious ways to deal with this, including the village irrigation system, which also I'm not going to talk into details about it, but for those who don't know, this is a water channeling system similar to the canal system that exists in many different parts of the dry world today. They also have adapted to the mountainous regions where most of the rainfall happens, including preparing these terraces for wheat cultivation, for example, or for other even perennial crops such as date palm. However, this is what's happening now. These areas have already been abandoned as a result of infrequent rainfall. On the right, there is the wheat terraces that are set in Musandam. Very few left of many of the mountains that used to be about one or two in Sia, maybe only one. And in the left, the ones in Jabal Akhdar, you would probably, if you visit there, hopefully you will have a chance, if you ever come to Oman next time, you will be able to see the terraces and how they are declining due to the decrease in rainfall in Jabal Akhdar. So this is not only happening in one part of the country, but also in different parts of the country. So these terraces and these irrigation systems like Aflaj have led to the biodiversity of all the crops that we can grow within a season. And also not only between crops, there is also diversity within each crop. So we have about 240 date palm cultivars, hundreds of other field crops and grain crops as well. So this has been sustainable for the longest time, in great part due to the invention of the foliage irrigation system, as well as modification of the land to become more growth. Now, what is our role in this day and age? Obviously, to preserve those resources, work on mitigating the impact of climate change, what actually reducing global warming and all the other impact of climate change, but also look deep into this and learn from what's going on, so whether it is agricultural system or it is the actual ecosystem itself, like this one in the desert. Because even though we are desert, as I showed in the first slide, we still have agriculture, we still have plants that can grow in desert climates. How can these plants grow in these kind of climates where it is totally no irrigation system and they are totally fine? They do away with their life cycle. And this is the actual work that we have been doing for the past few years. And so this is like, in summary, how the plant can modify itself to be able to grow in desert climates. And that includes using of the climate itself, including the mist, the dew, reducing evapotranspiration, the distribution of stomata, relative humidity, for example, making use of the water that's available in the soil as well as in the air. The soil as well play a major part in survival of these plants under desert climates, and our colleagues, especially Dr. Said Ismaili, is well familiar with the smart soils, and you'll probably hear about this in one of his talks that he is giving specifically on this topic. Soil fertility, water holding capacity, all these issues contribute to the plant survival in desert climates. But also the plants themselves. We have halophytes, we have xerophytes, we have all kinds of plants that can actually survive in the desert, and they do this through certain mechanisms that we haven't yet explored, especially in this part of the world, since we are in the less advanced in terms of scientific research in this particular area. But also there are other things that contribute to plants, and this is what we have been looking for, because we want the previous ones, whether it is a climate, soil, or plant, it is related to the species itself, to the plant itself. But what other factors that contribute to this, which is outside of the out of the plant itself? So what is outside of the plant itself? Then that's what we are interested in, and we are hoping that we'll be able to learn a few things from those. So we have looked at the microbes, we have looked at the soil mycorrhizae, fungi, bacteria, exophyte, as well as endophytes in this particular study. And so to sum it up, crop drought tolerance is in large part of these particular plants that are in the desert. When other plants are dying and they are surviving, is actually not have to do with the plant itself, although a major part of it, such as xylophyllum here, where the plant adaptation, as you can see, it is very short, widespread of roots, and the leaves have been modified to become very succulent, but also it has to do with other parts in the ecosystem, the soil ecosystem, and that is the endophytes. So now, if these plants can survive, why would the date palm die? And then we have to go and investigate what's going on there. And so that's what we have done. So basically, for those of you who are in a hurry, or if the internet got disconnected, this is what we have done. First, we went out there to the desert. We got them, isolation, identification of the endophytic fungi from these desert plants. And then we got them, knew what they were, and then we used those, including the one that is first record for the sultanate, which is Talaromyces omanensis, and a proof that enhances production, physiology, and anatomy of plants. So we stress the plants. In this case, we use a model as tomato, and then we were able to actually improve the yield, improve the growth of this plant using endophyte that were isolated from desert plants. So this is the story, basically. Now to the details. We're looking here at the role of endophyte fungi in promoting growth tolerance, and this is actually part of a thesis project done by my PhD students who are already now a doctor, Bushra Halo, and also in cooperation with my colleague, Professor Abdullah Al-Sadi. So what's happening here? Oman, despite being in a desert climate, still produces large number of fruit crops like date palm, banana, and limes, mangoes, and so many others, but this is the major ones. But also there is a whole bunch of grain crops like wheat, barley, as well as legumes like chickpea, and also vegetables in many different parts of the country, including onion, garlic, sweet potato, cucumber, watermelon, and tomato. And especially in cucumber, which is the largest greenhouse crop in the country, there's about 136 hectares of cucumbers grown, producing about 12,000 tons. And of course, the tomatoes, the major crop, major field crop in the country, with about 2,500 hectares, producing about 200,000 tons annually. This is according to FAO stat. So these are major crops. Tomato was our planned target crop because it is the main vegetable crop. And if we can work it on tomato, it can be applied to also to other crops. We have studied different aspects of this because these endophytes don't only contribute to the well-being or the water resistance or drought tolerance of these desert plants, but also contribute to their protection, as we found out that they can prevent certain diseases like Pythium, for example, from the damping of disease that leads to the loss of cucumber. But the plants themselves have different mechanisms that we have known long ago that are changing, or the drought stress affect a plant like tomato, for example. They affect the vegetative production, the reproductive life cycle of the plant, the physiology, the morphology, all aspects of the plant is affected by drought stress. So this is a major issue. Oman, as I mentioned, is already a dry climate. So that is a drought born, especially in tomato, because it is a field crop. Cucumber is usually a greenhouse that is using less water, but in tomato, the major part of tomato production in Oman is actually in the field, and therefore we have to deal with irrigation. We have to deal with natural synthetic growth promoter. These are some of the mechanisms to mitigate drought in plants. Nanobiotechnology now is going in, genetic engineering of these plants to produce drought-tolerant plants. Phytohormones, abscisic acid, for example, gibberellin can be used to enhance plant tolerance to drought, as well as microbial inoculation. And so endophyte is just one of these mechanisms that help the plant by activation of plant stress response system or through the biosynthesis of certain anti-stress biochemicals by these endophytes. So these are our goals, as I mentioned, isolation and identification of these endophytic fungi, as well as using these actually in a crop such as tomato and see how it can perform under water stress. So we went out, we used actually two different plants. One is called herm في عمان, or ratrat in some other parts of the world, and harmel, well-known plant also as well. Zygophyllum is the herm, and we isolated Cladosporium fungi from that. A whole bunch of fungi, not only that one, but this is the most relevant, but also Tularamyces from harmel. So these plants, when they live in the desert, like this one here, are doing that because of their association with these endophytes and not on their own. So this is the isolation and identification of these fungi. This is the procedure. I will not get into too much details on this, but we have done two ways. One is morphological description of these fungi, as well as genetic structure of these plants. So DNA fingerprinting basically of these fungi to match if they are something that we already know, or they are something that we don't know and they are new, and what kind of fungi that are associated with these plants in the desert. And we found out a whole bunch of plants, including aspergillus, a variety of species were there, as well as Saccharomyces, and our own samples were different isolate, as you can see here in this tree, than the one that already existed, which is mainly variables, Wortmanni, radicus, and many other types of Saccharomyces, especially those ones exist in Harmel. So after thorough studies and investigation of the genetic structure of this plant, we found out this is a totally new species that does not exist anywhere in the world and has not been reported before. And so we named it Cladosporium omanense. That is, of course, referring to Oman as our country. So the scientific name for this new fungi, endophyte, is called Cladosporium omanense. And so we were proud to actually have our country's name there. This is the identification of the fungi, the description, morphological structure of the fungi that makes it different under the microscope, as well as under the scanning microscope. And this is how the colonies look like for this particular fungi. I will not bore you too much with the details of this. But also on the other plant, we found not only Cladosporium, but also Saccharomyces omanensis as well. So this is also another species that is very distinct from other. You can see it here on the physiological tree here. That's the SKU Culture Center. That's our own culture there in the College of Agriculture and Marine Sciences. Also, this is a new species, totally new species that we found in Harmel, Razzia stricta. And so we have called it Saccharomyces omanensis as well. So both of these two species are referring to Oman. And this is how the fungi looks like under the microscope, as well as under scanning microscope here. That makes it very typical. And these are the colonies of this particular new fungi that is presented to the world from Oman. So basically looking out, and I'm sure and confident that the more we look, the more we find. And there are tons of different fungi out there that is waiting to be explored. And a lot of them are not found anywhere else, except in this particular part of the world, because this Oman climate, as I mentioned, is already a desert climate. Many of the plants have adapted to this kind of climate, and they didn't do it alone. I'm sure this is a fungi, but also a bacterial species, even viruses, despite the bad repetition of viruses these days. But they are very useful in mitigating climate change when it comes to drought stress. Okay, this is the first part. Now, the second part is now, okay, we found these two fungi. We're proud of it. We called it after Oman. So now what? Now we needed to continue and test it. Can we use this in a crop like tomato? And this is the second part of this experiment or this particular talk, because there were other parts as well. So we took this, and then we know that tomato grows in the field. We know it is subjected to drought. We have done so many studies on the effect of salinity, effect of drought of tomato in the college and with my colleagues, and all of that has been published and studies out there. So now, what can we do about it? This is what we are heading with this. So we took the fungal inoculated, the inoculation, we used it in 4 treatments. The first one, only the Saccharomyces omanensis without any drought stress. So what will happen if we inoculate these seedlings with this fungi without anything, without stress? Would that improve growth like it did in the desert or not? Then we introduced stress using polyethyl glycol at 6,000, 15%. And then we compare that to water. So these two treatments are drought stress and one with the fungi, and the other two treatments, one the control and one is the fungi without the stress. We look at all aspects of this plant, everything from pollen sterility to shoot growth to root growth, as well as the physiological indicators such as chlorophyll fluorescence and chlorophyll content. For those of you who are familiar with plants, these are very essential in determining if the plants are actually capable of carrying out photosynthesis, because that's the first thing that's get affected by drought stress. Even during the day, during the temporary wilting point or wilting of the plant, still the chlorophyll is largely affected. We also did some biochemical analysis of the leaves, including proline. When everyone also worked on plants and water stress, proline is a major compound that shows up whenever you have water stress, as well as we studied potassium concentrations in these plants. We did all analysis of the fruits, including the quality attributes, total soluble solids, tetrable acidity, the pH, vitamin C, which is ascorbic acid as well. And finally, we want to see how the plant reacts to the presence of this fungi, which is harmless as far as humans are concerned, but it is very useful for the plants. And we looked at the anatomy, how that will change the anatomy of the plant itself, because remember, these are endophytes that we are using, and we are inoculating the plant basically with a fungi, like we do with humans when we are waiting vaccine for coronavirus. We are doing that to the tomato, but in this case, to make it tolerant to drought stress. So basically, I'll go through the results really quick because my time is almost up. The pollen sterility of tomato plants increased significantly under drought stress. So that's whenever you stress plant, the first thing you lose is the pollen sterility, pollens. So there will be no pollination, no fertilization, and therefore there will be no fruits. And when we added inoculating the plants with the Tularomaceous omanensis, we found it enhanced pollen viability as well as pollen tube length. So this first major obstacle has been tackled using this fungi. Then looking at the growth characteristics of the tomato, it promoted some of the important ones, including shoot development, dry weight, and root length. So it did enhance the Tularomaceous, whether it is alone, like we see here in the first row of the table, or it is in combination with drought stress. All that has happened is that they were much better than when they were, of course, under only irrigation. So you can see here, shoot fresh weight was better, even though there's no stress, like under normal conditions. So this inoculation, or the fungi that inoculated the tomato, did improve the growth of the plant, despite the fact there was no stress. So even, of course, with stress, it did very well. So even those plants 113, for example, were significantly better than those under drought stress, even under irrigation. So when you compare a plant under water stress inoculated with this fungi, especially tomato here, then that will form better than another plant that is normally irrigated. It's not under water stress. So this tells you the role of these fungi in desert climates. So that's how it is. So they improve the vegetative structure of the plant. When we look at the fruits and the number of flowers and fruit weight and the fruit size, all of it did very well. As you can see here, the table or highlighted data shows that it did better than water irrigated plants. So this is interesting because having the plant inoculated with the fungi and stress them out will result in a better quality fruits compared than tomatoes that is normally irrigated and without any stress. And so this is how the data is, the fruit weight and the fruit size. As you can see here, this is the inoculated, and this is inoculated with drought stress, and this is irrigated regularly. And this is the one on the far right is the one that is subjected to drought stress. When we look at the physiology of the plant, the same thing is going on. Of course, we expect that because that improvement in the growth and that improvement in the fruit quality and the yield is a result of the improvement in the growth. And therefore, we expect that the physiology of the plant will be better, including chlorophyll fluorescence. That is very highly significant compared to those under water stress. And then chlorophyll content, whether you are talking about chlorophyll A or chlorophyll B. And these two types of chlorophyll actually harvest light at different wavelengths, light wavelengths. So it is very important that we study both and then come up with this conclusion. Carotenoid as a pigment is also very important, and as well as potassium and gibberellic. Even though it may not be significant, still it did perform arithmetically better under Tularomaceous omanensis. In terms of biochemical analysis, as you can see the results here, the treatment increase both, especially proline, potassium and proline. But proline is expected always to increase under drought stress. That was reduced. That means the plants were not going as much under stress when they were inoculated with the fungi. Obviously, those under irrigation and those under trichomonas. So it did not really. So the inoculation did not negatively affect the plant in terms of proline concentration. It did actually the opposite. Under water stress, there was a significant difference between those inoculated and those who were not inoculated. And then when we look at the quality parameters of the fruit itself, obviously we all know that all studies have been shown that under water stress, actually the plant would have higher concentration of total soluble solids simply because the fruit has gotten shrunken, smaller in size. And so the concentration of sugars in that would be higher, but that does not indicate higher quality because also other parameters will be higher, including the acidity of the fruit. And so when you look at one of the attributes that are very important for nutrition, which is ascorbic acid, we see that inoculated plants with the Thromyces omanensis were much were better, had more increased vitamin C compared to those plants that did not have the inoculation with Thromyces omanensis. In terms of anatomy, how this performed under water stress and without it, we can see that it changed cortex thickness and also external phloem thickness, and this is very important for the conductions of nutrients within the plant itself, from the leaves to the fruits to the root. And the xylem thickness has not actually changed much, but still better. And xylem physals, as well as the autolysis of the pith, which is disintegration of the pith. And so if you can see this better in the picture, this is how it looks like. Number one is the fungi. You can see the fungi makes the plant more conductive to nutrients and water in terms of phloem, cortex width, as well as a decrease in disintegration or disruption of the pith, as you can see it here in picture number 3 and picture number 4, because these 3 and 4 both were under water stress. Number 4 is the one that without any fungi, under stress but without the fungi. But you can see when we inoculated the plant and it was under stress, that the autolysis was much less compared to those under water stress. So even though the fungi is actually, let's say, it's a stranger to the plant, it did very well in terms of the plant health, into the reproductive tissues of the plant, the conductive tissue of the plant, both the phloem and the xylem, as well as the health of the stem. So in summary, we found out that these desert plants have certain interesting characteristics, including endophytes such as Thromyces omanensis, that provide multiple advantages to tomato under drought stress, including significant improvement in the plant physiology, in the plant vegetative structure, in the plant reproductive structure, in the fruit quality, as well as increased size and growth. And so this is basically the summary of the study. And what do we need to do next? And we need to just go ahead and use these as a valid drought stress mitigator agent that can be commercially produced in this country. And we can also explore, of course, further plans and see how can they tolerate water stress or drought under these desert climates. Look at the biochemical analysis of these cultures and do some drought enhancement impact of these fungi at the molecular level, because what we have done, as you can see, we have done the chemistry, we have done the morphology, we have done the anatomy, but we haven't gotten to the molecular aspects to understand the actual mechanisms by which these endophytes interact with the plants, resulting in all these changes. So these positive changes have grounds for them. We have not explored that at the molecular level yet. And so investigate the role. Now, what will happen if you combine it? If you combine more than one endophyte? Remember here, we only used one. There are omanensis in the tomato, just one. But we haven't used all these bunch of huge number of fungi that are associated, as well as bacteria and viruses that are present there in the root system, in the soil ecosystem, that provide these plants with the needed tolerance. But we focused on one, and this particular one can actually be synthesized into a nanoparticle, and they can be used in commercial scale. And this is not the first time. This has been done on other endophytes in other countries. For those of you who are interested, looking for more information, all this work has been published, and all aspects of this particular, like the two fungi, these two, Thelamensis omanensis, as well as Cladosporium omanensis, already there in publication, as well as the details of how these Thelamensis omanensis have enhanced reproductive physiology and anatomy of the tomato plant. In addition to that, we have actually done many studies, including those of the first time published actually here on the Bithyum suppression. Bithyum is a major disease of cucumber and tomatoes, and so using these fungi have contributed to that. So these endophytic fungi don't only contribute to drought tolerance, but also they contribute to disease tolerance as well. I would like to end with another study that my colleagues will talk about, which is related in a way. Now we have looked at the whole structure, microbial diversity and community composition. We have done that, my colleagues and I, in the field. Dr. Saeed Ismaili leading this project, and Dr. Daniel has investigated the diversity of this soils that are surrounding these plants that are under stress and under salinity stress and under drought stress, and found even the soil structure itself affected the presence of these microbial communities. And therefore, this also has been published, but there is abundance of functional microbes, including nitrogen-fixing bacteria, which are also considered and endophytes, nitrifying prokaryotes, as well as microbiota. All of this, especially in soils that are smartly designed soil, and that study will be, inshallah, talked about in coming presentations. But I wanted to link this up with upcoming presentation for those of you who are interested in the role of microbial diversity on the tolerance of desert plants to drought. And so we are hoping in the future that these plants that are surviving despite all these adverse conditions, including the heat, the salinity, and the drought, will be very useful in rescuing plants that are major crops in Oman, such as the date palm that you can see here dying in Sneina. This picture is from Sneina. And so we hopefully will be able to do that. I thank you for your time. I apologize if I got too long. And thank you. I appreciate your support and invitation to talk on this presentation. I'll be happy to answer any questions you have. Thank you, Dr. Athman.
Thank you very much, Dr. Rashid. I appreciate the nice presentation. And please be patient with us. I would defer the question